Hello, in this video we're going to talk about the centrosome, the cell wall, and the extracellular matrix. So let's not waste any of your time. Let's get right to it. Now when we're talking centrosome, we're talking its big task occurs during mitosis, and we referenced this when we were talking about microtubules earlier. But you can see the picture there beside me. The centrosomes almost look like little earplugs, but anyway, they are the organelles. Thinking tow truck. Okay, I think referenced this in the earlier video on cells. But it's the idea that tow truck dragging around, in this case, chromosomes, using the microtubules is kind of like a chain. But just the idea of having that process of moving stuff around. Of course, everybody, individual one is a centriole, pair of them is centrosome. But it's this idea of organizing, moving things around, and getting them set up for that mitosis phase. Okay, continuing thinking about centrosomes, got a pretty tough, durable structure, as you can see, hollow. Okay, except for the one rod in the middle, and the going around. You can see they're made of microtubules, and they're sets of three kind of bonded together in their own little sleeve. And so if you think about a straw, maybe with spaghetti, yeah, you could take spaghetti noodles, uncooked of course, and put them in groups of three and kind of glue them to the straw, kind of that kind of structure, but pretty strong, pretty durable. Little old organelle. Okay, now we're talking eukaryotic cell walls. We spent a little bit of time talking about bacteria cell walls back in our introductory video, and how tough and durable that is. But a cell wall for eukaryotic, which mostly be plants and again fungi, some protists, much different than bacteria or prokaryotic cells, which are almost indestructible at times, or sure it seems that way. But for plants and protists, it's made of cellulose, and if it's a fungi, it's made of chitin. Chitin we referenced in the macromolecule videos. That's that whenever you step on an insect and you get a little crunch, like a beetle or a cockroach or whatever, that's the chitin breaking. But cellulose, cell wall for plants, like you take some celery and snap it in half, and you get those strings that are hanging down. That's the cell wall. That's the cellulose that you're seeing there. Okay. Now again, the cell wall, just like it is for bacteria, support, protection, shape of the cell. And I mentioned a couple of times the relationship between the central vacuole and the cell wall and the turgor pressure. But it does a good job of just keeping it the shape, and it's also part of the protection. One of the things that our cells have to worry about, like for example, with homeostasis, or even going to a doctor to get an IV or something, it's the idea of, and we talk about this in, is it osmosis? Yeah, so chapter 5. The idea of osmosis, water moving in and out. Our cells have to be balanced, because if they get too full of water, they could burst. They get a neglect of water, they could shrink up and die. But a cell wall for a plant helps to prevent that. I mean, because it likes lots of water. It likes to be a full, lots of pressure, so it can stand upright very efficiently. So that cell wall protects it from bursting, okay, or even shrinking as the pressure changes. Oh, excuse me. And like when you're eating veggies, carrots or pickles, peppers or whatever, that crunch that you hear, which is a good part of a sandwich, that's the cell wall being broken. Now, in this next part, plants have cell walls, bacteria have cell walls that do a good job at protecting the cell. Okay, ours doesn't have that, but that doesn't mean that we don't have something with that in mind. Okay, we do, and that's the components of the extracellular matrix that we're going to talk about now. And this probably is our longest section. I think it's got quite a few slides tied to it. But what we're talking about is this stuff around the membrane. Okay, so you got the membrane, and if you're thinking that was the outer portion of the cell, that's not necessarily the case. The extracellular matrix, it's things that it's part of the organelle process, but they're actually outside of the cell, attached to it, with the idea of doing what it can to protect and to help with that. You can kind of see in the picture beside me that idea of being on the outside, and a lot of collagen, a lot of fiber stuff, and we'll talk about each of these in their own particular slides, and you can see the proteins there, the polysaccharides, collagen, a lot of things going on to protect. And kind of think of it as a cytoskeleton on the outside. And it can also influence cell behavior. There's some communication involved. There's some ability to exchange materials between cells. So we're going to look at all those. Okay, one of the ways, as I just mentioned, that this extracellular matrix is a benefit is cell-to-cell interactions, whether it's information, whether it's materials. Some of it's going to seem like, if like, for example, if you built two houses and you built each one of the walls on each house right beside each other, with just an inch or two difference, which you would never do, and I take that back, maybe in some areas that's a necessity because of space, and you put matching windows in the exact same position to where I could open the window of one house and hand you something in a person in the other house. That's how close they are. We're kind of thinking about that. The surface proteins can give the cell identity, or can even be used to recognize other cells and what's going on. But cells that are close by can read and react to each other. There is some communication. There is a situation where if a cell gets infected by a bacteria cell, it sends out a message to that extent, and the other cells lock it down. Borders up, shields up, all that stuff. And so there is some of that. As we mentioned, the glycolipids being readers, recognition of foreign material, and all that stuff, and that's all part of the immune system. It's also part of the process that transplant patients have to go through to try to reduce that reaction, because if they get a new organ like lungs or whatever, they don't want their immune system to destroy those cells, because the immune system is going to say, hey, those cells aren't us, they're not us, we got to get rid of them. And so it's that process of working through that. continuing to think about the extracellular matrix when it comes to cell connections, whether it's an exchange of information or materials, whatever it may be. There are three types. There's your tight junction, and again, each of these will have their own slide that we will talk about. But you got your tight junction, and that's just what it says. It's an idea of information exchange, but not materials. No leakage, no materials. It's super tight, and again, but we'll look at that. There's an anchoring junction. Okay, there's also a communication junction, and the communication junction is what I'm going to reference again, the two windows, and even more than that. If we put a tube in between the two houses, to where we can just talk directly into it. But that's the idea of the three types. But again, they each have their own slides that we will get to. Okay, now when we're thinking tight junction, we want to think almost like brads on a jean, if you know what that is. Those little brownish red parts of the jeans that are in the stitching that design to hold things together. This picture beside me, that's a portion of the membrane of two different cells, and you can see they're ratcheted together. They're side by side. Maybe it's a muscle tissue, maybe it's a brain tissue, maybe it's skin. Whatever, but it's the idea of connecting them together, so you can have that communication. And again, it's called no leakage because there isn't chemicals or it's just information. The idea of being that connected together, but that's tight junction. Okay, this next one, anchoring junction, or a decimone. You can see that again, the blues. There's two blue layers. Those are two separate cells. The membranes are two separate cells. But now, instead of and the wavy things, those are interfilants, inside the cells. But now, instead of having those brads, now we're going to be anchoring like spikes on each one that connect the cells together. Those intermediate filaments essentially push through, and the long parts on the inside and then the short little part on the outside of one, and you got the adherens of another. Those connect together to kind of make that connection very strong. I'm almost thinking of it as spot welds, welding something together. That's decimones. Okay, our next one to think about is plasdecimone. These are the communication junctions. There's two types of communication junctions. We'll look at one here, and then our final slide will be the last one. But these are channels, openings. Okay, you can see it in the picture. I've got two plant cells in the cell walls, and there's just simply an opening between the two cells. Allows material to pass through. Information can pass through. Chemical, electrical signals can pass through one to another. But it's just the idea of having that opening between the two. And that's got obviously good points. It's obviously got bad points. You have an opening in the cell wall, but the opening is only in such a place to where the two cells can access it. A bacteria cell couldn't access that opening because it's up against the side. I mean, those two are connected. Bacteria cell would only have access above and below, but not that general opening. Now the next one. Next communication junction is the gap junction, And this, again, you can see the two separate plasma membranes of two separate cells. Now this one is a unique one because its structure is basically 6 proteins working together to build a kind of a tube. Now, whereas the previous communication in the plasmamonas, those were always open as just a hole. So if you can imagine, if you just in your house, you got those two houses side by side, you just knock holes in the wall so you could talk to each other. That'd be one way of doing it. Or, as I referenced, a window that can be open or closed. These gap junctions can be opened or closed based on need, and therefore these can be anywhere on the cell. It doesn't have to just be, butted against the next one, so bacteria couldn't access it. These can be opened or closed, but they're basically tubes between two cells, just like if I had 2 windows in 2 neighboring houses that are really, 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 really close, and we open up the windows and we put a tube that completely filled the windows, and then we had maybe a lid or a cap on both ends where we could open it, we could close it based on need, but that is a gap junction. Well, that is our video over the extracellular matrix, and we also looked at I've already forgotten. Cell wall and the centrosome. It also is our last video for Chapter 4, the organelles. So next time we get together, we'll be in Chapter 5, plasma membrane. I love that chapter. So we'll see you then.